Hello, students. In this lecture, we'll talk about the screening of recombinants. The success of an RNA technology lies with the identification of the recombinants. So, selection and screening forms a two important methodology for identification of this recombinants. So, we have already discussed about the selection methodology in another video. So, I recommend you to watch my another video on selection of recombinants. The link is given in the description. So, this selection involves methods like visual screening, insertional inactivation, complementation of mutation in the case of yeast vectors, the reporter gene assay in the case of eukaryotic vectors and cell lines. So in this video, we'll be discussing about the methods which involve the screening. That is, the screening of recombinants or the identification of the recombinants from a host of cells, which contains both recombinants and non-recombinants. So it is a selection of a particular cell, identification of a particular recombinant from a library of clones. So the methods that we'll be discussing in this video will be colony hybridization, the blotting techniques, and colony PCR. So this colony hybridization methodology was developed by Costin and Hogness in the year 1975, and it requires a radioactively labeled DNA probe with a sequence which is complementary to at least one part of the desired DNA. The probe may be partially pure mRNA, a chemically synthesized oligonucleotide, or a related gene, but which will identify the corresponding recombinant DNA. So the basis here is the complementarity between the probe and the sequence which has to be identified. So this is a methodology. So the colony to be tested are replicated from a solid medium onto a nitrocellulose filter paper. The paper is treated with NaOH, that is, alkali lysis, occurs here, which will simultaneously break open the cells and denature the DNA. Now, while we are transferring this colony onto a medium to the nitrocellulose filter paper, a portion of each colony will remain on the medium, and this will be known as a master plate. And now on this nitrocellulose filter paper, we are carrying out this alkali lysis. The paper is then saturated with P32 labeled mRNA, that is a probe, which is complementary to the gene being sought. Now, if there is complementarity between the desired DNA and the probe RNA, what happens? Now they will form a hybrid. After washing, remove the unbound mRNA, the probe. The position of the bound radioactive phosphorus is detected by the autoradiography. Now, if you are looking for the protein product of the gene, then you'll be using immunological techniques like it will be an antigen antibody interaction to identify the desired gene, the desired protein in this case, right? So these are the steps which are involved in your colony hybridization. And we have the illustration of this colony hybridization over here. So as you can see, you're having a plate and a nylon membrane. So the nylon membrane is placed over the nylon membrane is placed over the plate, and this plate, the nylon membrane is placed over this plate, and the colonies are transferred, and we find that a few colonies are retained, and this makes up the master plate. Now once they are transferred onto the nitrocellulose filter paper, this filter paper is again placed on a membrane like a new agar. Now this will have the colonies. You will denature and neutralize and wash. So the denaturation is carried out with the help of an alkali. Now what happens is the bacterial DNA is fixed on the membrane. Now you will be labeling it, hybridize it with the labeled probe. It might be an mRNA or a DNA. Now, if at all there is complementarity, as in this case, you can find there is a hybridization occurring, which can be detected by a autoradiography. Now, compare this autoradiogram with the original master plate, and you can pick up the colony, which is exactly in the same location. So, these represent the recombinant which you desired for. So later this can be manipulated further. Now, the following methods will depend upon nucleic acid hybridization, the blotting techniques. We are going to look into the blotting techniques. Now, these blotting techniques are widely used as analytical tools for the specific identification of either a DNA or RNA fragment from large number of molecules. So, the blotting is actually referring to the process of immobilization of the sample nucleic acid on the solid support. Okay. So literally you are immobilizing the nucleic acid either via DNA or an RNA onto the solid support, which is a nitrocellulose or a nylon membrane. Now, after blotting, the blotted nucleic acids are then used as target in the hybridization experiment for their specific detection. So here again, the hybridization techniques or the blotting techniques depend upon nucleic acid hybridization, that is the exact complementarity between the bases of the desired DNA, that is your target DNA, foreign DNA, to that of your probe. So in that case, the probe will be having a base pairing, and once it binds to this target, you are going to identify them. So the first method that we'll be discussing is Southern blotting. So this method was developed by Southern in the year 1975, and he was analyzing the related genes in a DNA restriction fragment. So apart from this, you can this technique is utilized to provide physical map of restriction sites within a gene. So the mapping within a gene with the help of the restriction sites, which is located normally on a chromosome. And it also reveals the number of copies of the gene in the genome. And it can also be used to identify the degree of similarity of the gene when compared with other complementary genes. So this is a schematic illustration and the steps which are involved in your Southern blotting. So let's go through this. So the first step, you find that the DNA is cleaved. So from the host, the recombinants and non-recombinants from the library of the cells, library of clones, you are obtaining the DNA and you are subjecting this DNA to agarose gel electrophoresis. So since the DNA are larger fragments, you can go in for a step which involves the fragmentation will be the use of restriction enzymes. So you can go for a restriction digestion of this DNA and then you go for a agarose gel electrophoresis. After separation, you can run a marker side by side so that you identify the size of the fragment which you are obtaining. Now, these fragments from the gel, the DNA fragments from the gel are just blotted onto a nitrocellulose membrane, the filter, nitrocellulose filter or your nylon membrane. So here in this case, nitrocellulose filter in this diagram. So what happens is the nitrocellulose filter will now absorb, the DNA goes and adsorbs onto the or gets immobilized on the nitrocellulose filter. So to make the cross-linking of the DNA onto the cross-linking of the DNA onto the membrane, what you do is you subject this to a temperature of 80 degrees Celsius at the 2 hours, 1 to 2 hours. And this process is known as baking. So baking will allow the cross-linking of the DNA onto the nitrocellulose filter. So and alternatively, you can also use UV. So once you have transferred this DNA and immobilized this DNA on the nitrocellulose filter, now what you're going to do is now when you're doing this, one important step which is involved in between is the lysis, alkali lysis. Since the DNA here which you have separated is double stranded, when you have the one which you're transferring onto the nitrocellulose filter has to be in the form of single stranded region. So that later when you're going to add a hybridize with the probe, the identification is made possible. So it has to be converted into single stranded DNA, which is achieved by alkali lysis. Now once you are transferred the single stranded DNA onto the nitrocellulose filter and allowed a baking procedure or UV cross linking so that there is the strong binding of this DNA onto the nitrocellulose filter. You go for the next step, that is the addition of a radioactive probe. So this radioactive probe can be a DNA or RNA. So here this you are adding a homologous DNA. So once you have added, later you allow the appropriate incubation time, and then you're going to wash the unbound DNA probes. Then this filter is exposed to the photographic, the autoradiography. You go for the autoradiography procedure. So now you have the darkened bands. So the bands were the one which representing the hybridization of the labeled probe, radioactive labeled probe, and the desired DNA which is present on the agarose gel. Now you compare this autoradiogram with the original gel, and you can identify that your recombinant, the desired gene. Now, this diagram shows you the elaborated version of how the blotting actually occurs. Why this technique is actually known as blotting is, now this is the methodology by which you are set up. You call it as a southern blotting, the set up. So how you do that? So you have the buffer in this trough, and later you are going to provide a support, and then you are providing paper wick. You place the agarose gel over this, then next followed by the nitrocellulose membrane, and above this membrane you are placing the Wattman, a stack of Wattman filter papers, 3 paper, and then paper towels. And above all this you place the weight. So the actual mechanism over here is there is a adsorption of this buffer from the reservoir by this filter paper stack. So when there is adsorption due to this capillary action, there is movement of this buffer, the adsorption of this buffer by this paper wicks and the Wattman filter papers. So when this occurs, DNA is also drawn from the agarose gel, and it is now stuck onto the nitrocellulose membrane. So it gets blotted. So that is why you call this technique as a blotting. Now, since your Southern blot, Edward Southern utilized this method for DNA, we call it as a Southern blotting. And the next method which Alvin discovered was identifying the RNA. Since it was an extension of the method of Southern blotting, it was known as Northern blotting. So Alvin devised a technique in which RNA bands or blot transferred from the gel to a chemically reactive paper. So here, please note, instead of your nitrocellulose membrane, there is a chemical reactive paper, that is DBM paper, diaminobenzyl oxymethyl cellulose, DBM paper, a chemically reactive paper. So the hybridized bands are found out by autography, similar to that of your southern blotting. So here in this technique, the blot transfers are reusable because of the firm covalent bonding of the RNA to the reactive paper. To nitrocellulose paper and then hybridized by single standard probe, which is either RNA or DNA. The hybrids are then treated with S1 nuclease to remove the single standard non-hybridized probe, which helps to you to quantitatively estimate the mRNA. So here, now we have seen that this nucleic acid hybridization technology, you have two methods. One is the southern, the other is the northern. Southern blotting, you have the immobilization of DNA on the nitrocellulose membrane. Northern blotting, you have the immobilization of RNA on the membrane or the reactive paper. So the identification can be either by DNA or RNA probe, but essentially southern blotting leads you to the identification of the recombinant DNA, desired DNA, and northern blotting allows you the identification of the transcript, that is the RNA. Now, Western blotting. So this method was developed by Taubin in the year 1979. So it is like a method which is developed to detect the protein, that is the gene product. So please make a note that southern and northern blotting, we were identifying the either the gene or the transcript. So here identifying the gene product, that is the protein. So when the transferred gene expresses a protein in an alien cell, then it is detected through this technique. So here you are going to electrophorese the protein by means of phage. So after you do that, you blot it onto a nitrocellulose membrane, right? And this hybridization of proteins is done by radio-labeled antibodies. So this is like a more like a immunoblotting, antigen-antibody interaction. So you can also refer to it as a immunoblotting. And this again, if it is radio labeled, you go for autoradiography. You can have variation of the methods. Now you can also use fluorescent tagging or enzyme substrate reaction, so that you use a secondary antibody which is tagged with an enzyme. The primary antibody will be like having a substrate link. So here, this is the illustration which shows you the Western blotting technique. So the antigen, you can see the antigen samples are separated, right, on a membrane, then transferred onto a membrane on a gel, polyacrylamide gel, then transferred to a membrane, right? And then you have the labeled antibody. So here in this case diagram, we show the detection by means of fluorescence. So it can be chemiluminescence or it can be your enzyme substrate linked. So in that case, you'll be using a secondary antibody. So you go for the identification if it is through autoradiography. Now, dot plot hybridization is again a nucleic acid hybridization method where you are going for spotting the DNA, right? So you're extracting the DNA, genomic DNA, and you're spotting it onto a nitrocellulose membrane. So it where you are going to use an apparatus is used for placing the spots on the membrane through slots. Thus, the spots are made in the form of oblong slots rather than round blots. So you have this either dot blot or slot blot. Okay, so this step does not involve digestion with enzyme, gel electrophoresis, or the transfer from gel to membrane. So just a slots or the dot spots, the dots that you have it on the membrane and you go for. So multiple samples are being identified at a single screening. So this technique is considered much more convenient when no restriction sites are needed to be studied, and only it detects the presence of foreign DNA clone in the cell or the tissue. So here we have the illustration. So as you can see, the DNA, step one, the purified DNA or RNA at different samples. Step two, you are applying them directly as a small dot. So this is a dot blot assay. You're having the dots being applied onto the nitrocellulose membrane. You go for baking, right? And then step three, you add the labeled probe for hybridization and incubate. Step four, wash off the excess bound probes, unbound probes. And then step five, since it is radioactive labeled, you go for autoradiography for identification. So screening can also be done by PCR. So PCR has become as a significant technique in identifying and isolating positive clones from the libraries. So the sensitivity and specificity of this PCR technique is well much better than the other methodologies. So these are the advantages. You eliminate the false positive clones. Since only positives are identified by DNA bands of appropriate size, the overall screening time could be shortened and multiple genes can be screened with appropriate primers. So, an example of this methodology is given here, right? So, it is a convenient method. So, what happens here is that we know that PCR, there are three basic steps, like you have the denaturation, annealing of the primer, and then your extension. So here in the initial heating step, this will cause the release of the plasmid DNA from the cell. So it can serve as a template for the amplification reaction. Now the primers are designed which are specifically targeting the insert DNA, the foreign DNA which you have inserted into the clone, right? So your primer will be designed which is specific against this, and so they can be used to determine if the construct contains a DNA fragment of interest. The insert specific primers can provide information on both the specificity and size of the insert DNA. While the use of vector specific primer, you can also utilize another type of primer which is like specific to the vector. Now, this will allow you the screening of multiple constructs simultaneously. Now, when you are using an insert specific primer, it is highly specific, right? And if you're using a vector specific primer, multiple like uh, cells can be screened. So, where irrespective of whatever the experimental design be, the presence or absence of a PCR amplicon and the size of the product are determined by electrophoresis alongside a DNA size marker on an agarose gel. So, As a result of this rapidity and the sensitivity of this method, nowadays PCR is the most preferred method for screening the large genomic libraries or the cDNA library. So with this, we conclude the methodology for screening the recombinants from a host of the clones. So these methods that we have discussed include the colony hybridization, the blotting techniques such as northern, southern, and then western. And to conclude with, we have discussed about the use of colony PCR for identification of the recombinants. Thank you.